Hello, thank you for coming to my presentation. My name is Yeremia Gunawan Adhisantoso, and I am a PhD student at Institute for Information Processing, Leibniz University, Hanover, Germany. It is a pleasure to give a talk about my research topic titled PEKORA High-Performance 3D Genome Reconstruction using Gate Order, Spearman's Rank Correlation Approximation. Before we start with the 3D genome reconstruction, I will go through the basics of 3D genome structures. I will start with the hierarchical structure of the genome. Our genome is densely packed inside the nucleus, yet it creates hierarchical structures or regions that serve certain functions. So as we can see here, it's not randomly packed inside the nucleus. First, I will talk about the chromosome territories. Gene-rich chromosomes tend to be located at the center of the nucleus, while the gene-poor chromosomes are located at the outer part of the nucleus. Furthermore, chromosomes is partitioned into A-B compartments. A compartment or open chromatin is a gene-rich region, and B compartment or closed chromatin is gene-poor region. And then as we can see, it's also furthermore partitioned into so-called loop domains and ordinary domains. So why is the 3D genome structure is important? So if we see our genome in a linear way, it's quite often that we see that the enhancer and the promoter of a gene are located far away from each other. So if we see the locus of the enhancer and the promoter, the distance are quite far away. And it turns out that the enhancer and the promoter in the nucleus forms a so-called loop, where the enhancer and the promoter in terms of spatial distance are quite close from each other. This model is called loop extrusion model. In order to reveal the interaction between enhancer and promoters, and also to reveal this kind of structure, the structure that I already explained earlier in our genome, we do the so-called chromosome conformations capture. Here we call it 3D genome sequencing. First, loci or region of the genome that are close with each other are first captured by cross-linking the DNA. And then the segments are cut using restriction enzyme. And then the ends of the segments are marked with biotin. Those parts are then ligated and then purified, which then at the end of the process sequenced using pair-end sequencing and then aligned to the reference genome. So for example, here on the left-hand side, we have blue and orange part that belongs to different segments. We find through the alignment, we find the positions in the reference genome. So assuming that the blue part are located at the positions between 30 to 39, and then the orange part is located between 20 to 29. So we create a matrix called contact matrix, where we add a count 1 to that specific loci. The loci are then partitioned into a certain size called bin size, or furthermore it's called resolution. Each entries of the contact matrix, we called it interaction frequency. Important here is that this kind of interactions correlates with the spatial distance through the process. So for example, if two different loci are located near with each other or spatially near with each other, the counts will be higher, and where the loci that are quite far from each other, we will have lower values or even no values at all. But this process is imperfect, or it's noisy, so of course we have noise in the contact matrix too. At the end of the process, we reveal the structures using, for example, 3D reconstructions. So what are the challenges in the contact matrix and 3D reconstructions? First is that the contact matrix contains large amount of missing data, and the observations tend to be noisy. On the right-hand side, we can see that there are regions that are marked with blue. Those regions doesn't contain any observations at all because those regions are non-alignable. The centromeres is also not visible in the contact matrix. The number of observation is less than much smaller compared to the n squared, where the n squared is the number of loci. The next is we have the problem with scalability. As I already explained earlier, that the genome contains hierarchical structures, and then in order to reveal, we requires high resolution data, or the bin size must be smaller. The problem with high resolution data or smaller bin size, that means that the contact matrix grows quadratically. In result, our method grows also quadratically. And last is the problem with low accuracy prediction across chromosomes and cell types. Here, in our work, we focus only on intrachromosomal contact matrix, or that means the contact matrix which describe reveal the interactions between the chromosome and itself. So now I will explain about the 3D reconstruction pipeline and how the performance is evaluated. As the input, we have the contact matrix C. And earlier, we already talked about that the interaction frequency is inversely proportional to the spatial distance in 3D space. Therefore, the contact matrix can be converted into Euclidean distance matrix, or into a so-called Wish distance with the conversion factor alpha. Furthermore, in the Euclidean distance matrix, we set the diagonal into 0 because the Euclidean distance between a loci and itself is always 0. Now, the loci is modeled as bits of string. For example, here it's represented, the position is represented as the matrix P with size n by 3, where n is the number of loci. Furthermore, we have partial observation, so only entries at the coordinate ij, that is which is element of omega, has an entries, non-zero entries. The number of observation is much lower compared to the n squared, and in order to compute the Euclidean distance matrix, first we compute the Gramian matrix, which is the product of P and P transpose, and then at the last, we compute the mean square distance between our observation and the predicted distance, Euclidean distance, based on the loci position. After the optimization is complete, we have the Euclidean distance matrix, which is it is then reverse converted into the so-called predicted contact matrix. The similarity between our data contact matrix C and predicted contact matrix is then measured using Spearman's rank correlation. In the past few years, progresses have been done in the field of 3D genome reconstructions. Here I listed 3 state-of-the-art methods. The first one is SHNATE, which models using the multidimensional scaling methods, and it models the neighborhood relationship between loci. And then it also controls the smoothness of 3D structure using distance penalty. Two other methods, hierarchical 3D genome and Flamingo, tries to solve the problem by first dividing the chromosome into, the contact matrix into domains. For example, hierarchical 3D genome divides the chromosome into smaller domains, where the domains are predicted based on arrowhead algorithm. Furthermore, it uses additional objective functions, Lorentzian objective function. On the other hand, Flamingo, instead of using external information, external domain information, it divides the contact matrix into equal-sized domains. And for the objective functions, it uses a rank constraint and maximum distance constraint. With the maximum distance constraint, it is similar to the SHNATE. So now I will talk about our method on how we improve the current state of the art. So our method PEKORA is built based on 3 methods. The first one is we optimize it using the so-called alternating minimization, which is non-convex optimization. And then in order to optimize it, we use gradient descent based methods called ADAM. And then furthermore, we use Berzeli-Borwein methods to adapt the step size at every single iteration. And then at the end, we add, similar to other methods, we have additional objective functions which work as a regularization. Here it's called kth order Spearman's rank correlation approximation. Let's start with the alternating minimization. The Gramian matrix scales quadratically to the number of loci n, and the state-of-the-art methods keeps the whole Gramian matrix in the memory, which leads to the quadratic memory complexity. Next is that with the rank constraint, it can lead to the lower dimensional space solution, which discovered by Chris-Lock et al. On the other hand, we try to only optimize based on the observed data. Here, for example, we have the position of the loci, pi for its loci, and pj for the jth loci. And then, for example, when we compute the error, we take the difference between our observed compared to the predicted Euclidean distance. The problem here is that it is not convex. Therefore, in order to update pi and pj, we do it separately. For example, to update pi, we consider that pj as a constant, and at the same time, to update the pj, we update the pj by taking the gradient of the eij and making the pi constant. To optimize alternating minimization, we use gradient descent-based method ADAM, which is first-order derivative method, combined with Berzeli-Borwein method to estimate the step size at every iteration. For a second-order derivative method, for example, here second method, it approximates the Hessian matrix based on the gradient of the current iteration and the gradient of the previous iteration. But instead, with Berzeli-Borwein, we don't approximate it as into a matrix, but instead into a single constant. Of course, a single constant cannot approximate one full matrix, and therefore we have residual. And then in order to minimize, we compute with the squared error. But because it is squared error, we can compute the minimum point, and then we can estimate directly the constant. So now I will talk about the kth order Spearman approximation. Assume that, for example, our data are sorted in ascending order and the corresponding prediction are sorted accordingly. We can reformulate the Spearman rank correlation into the ones that's shown below here. So the rank of dl or the rank of dij becomes the index L because it's already sorted, so the rank at that position is equal to the index. And then to compute the Spearman rank correlation, we only need to compute the rank of our prediction. In order to improve the Spearman rank correlation, we need just to focus on the numerator here because the rest are constant. Spearman rank correlation is inherently not differentiable. And therefore, we try to approximate it based on three observations. On the right-hand side, we can see that, as I explained earlier, that the predictions are already sorted according to the sorting of the data. First, we start with the first point or the point at index 1. The rank of the first point is correct because the remaining points, other points has the value greater or equal to the first point. So in this case, for the first point, we don't have to penalize the value. Next is the point at the index 2. We have to adjust the prediction because the points at index 3 is lower compared to the point at index 2. That means the point at index 2 has a rank greater than 2. How we improve the prediction is by reducing the values of the second index and at the same time increasing the values at the value at the index 3. The amount of how we increase or decrease is based on the difference between 2 points. This doesn't violate our first observation because we always observe the relationship between 2 points with offset O. Because we have two different interactions for relationship. For example, the first one is that as long as the next value is greater than the current value, our loss is 0. But on the other hand, if the next value has the value lower than the current point, we use non-linear functions ReLU here. So we only penalize when the next the point with certain offset O is less than the current point. Last is that where if we compare every single prediction with other possible predictions, we have n to the power of 4 complexity. Therefore, we limit only to a certain offset, and therefore we have this kth order Spearman rank approximation. For the experiment, we evaluate the performance based on the data set from Rao et al. In 2023. This is the data set commonly used also by other state-of-the-art methods to measure the performance. The dataset is based on the cell line GM12787 at 5 kilobase resolution, and it contains the contact matrix of chromosome 1 to 22. For the PEKORA parameter, we use the conversion factor alpha 1.0, and empirically, we found that order of Spearman rank approximation 10 and width for the regularization 0.05 are sufficient to achieve good result. So first, we start with the memory consumption and the runtime for the chromosome 22. Here we measure the maximum memory and the runtime of our method in comparison to the state-of-the-art methods. In both cases, we outperform the state-of-the-art methods as our method doesn't scale quadratically to the number of loci. And with the ADAM and Berzelius-Borrow method, we can converge much faster. Here we can see that it requires only 3 minutes in comparison to the other state-of-the-art methods, while achieving better performance. For we measure the performance for all of the chromosomes. So here we have the result for chromosome 1 to 22. And on average, we are 30% better compared to the hierarchical 3D genome, which is the current best state-of-the-art method. To summarize my talk, I introduce you our method PEKORA, high performance 3D genome reconstruction using kth order Spearman rank coordination approximation, which is designed around alternating minimization, ADAM and Berzelius-Borrow, and also additional objective functions called kth order Spearman rank correlation. It achieved on average 30% improvement over hierarchical 3D genome, which is the current best state of the art. For the outlook, we already measured based on the objective matrix, but now we want to compare or we want to measure the reconstruction accuracy using the data from different experiments, for example, SPRITE and SHAPET. Furthermore, we also want to measure the performance based on the so-called HiTSeq reproducibility matrix, Genome Disco, and HiTSeq Rep. Thank you for your attention, and see you next time.